Good afternoon. My name is Dr. Sanjay Suresh, and I'm a student taking Immunology 1060 at University of New England. So today in my video, I'm going to be talking about immunoassays. So what is an immunoassay? Immunoassays are a biochemical test which measure the presence of a substance in a solution by using antigens and antibodies that bind to it. So today in my video, I'm going to be talking about three major immunoassays, which is the ELISA, and the radio immunoassay, and the last one is the fluorescence immunoassay. So what is ELISA? Well, what ELISA does is it detects the presence of ligand in a solution by using the antibodies which bind against these ligands. So this creates a reaction, and it can be used to detect various diseases. For example, Lyme disease, HIV, Rocky Mountain spot fever, and also we can use it to detect various autoimmune diseases like SLE, Graves' disease, thyroid disease, etc. So how is this ELISA test normally done? Well, it all starts off by the antigens. So what they do is they add the antigens. The antigens are added to the surface of the cell, and these are mainly bound, and these antigens have to match the antibodies on the surface of the cell. So once the antigens and antibodies are bound, these are linked by an enzyme. So this is mainly linked by an enzyme. And once the enzyme gets linked, any unbound antibodies are removed. And to this antigen antibody enzyme linked complex, we add a substrate. And once the substrate is added, we see a color change. So if we see a blue color change in the ELISA test, it indicates that ELISA is positive. So all the antigens and antibodies have been bound. If there is no color change, then this indicates a negative ELISA test. So this is the main principle, and this only involves one antibody. So this is the main principle of ELISA. The second thing that I'd like to talk about is the radio immunoassay. And this radio immunoassay is a very sensitive test, and where we measure the concentration of the substance by using the sample of the antibodies. So this can be used to detect various hormones, enzymes, and also drugs. And the type of substance that is used is the gamma radioactive iodine, that is 125I. So what do we do in this radio immunoassay? Well, we use a protein which is mixed with the antibody, and this protein and antibody which is mixed is specific to the protein, and later to the sample an antigen is added. So once this antigen is added to this protein antibody substance, this antigen typically it has to compete with the labeled antigen, and once the label antigen is competed, it binds to the antibody, and any bound or unbound antibodies are separated, and later the radioactivity is measured. So and this can be used to measure any hormones and enzymes, and it's a very common technique which is used as the immunoassay. And the third one that I'd like to talk about is the fluorescence immunoassay. And here we use the light microscope, which allows detection. And here we localize the target ligand with the target ligand molecule within the cell or the tissue, and we measure, and this is measured at a quantitative level. So what happens here is that we conjugate the antibody with a substance called as fluorophore, and it is used to visualize it's used to visualize the location of the target molecule. And this is a very important technique used in immunostaining and immunohistochemistry. And this is also widely used across. So the three major immunoassay techniques that I've talked about is, one is the ELISA and one is the radioimmunoassay, and the other is the fluorescence technique. And by using these techniques, we can diagnose various different diseases, infectious diseases, and also autoimmune diseases. So this is it for my video today. I hope you like my video, and please do subscribe to my channel. Thank you.